Thank you, Noel, for inviting me today. It's a pleasure to be at Google and actually see the campus, first time for me. And today, the talk is going to be about, there's really twofold in the talk. I'm going to try to bring up the technology and the type of thing we've been involved with that links well with palimpsest and actually Google's technology. And at the end, I will try to link it back to actually the type of work we've been doing and show you where those needs could play a role. They don't play a role yet, but and I will show you a bit about how we do those things. So before I go into the biology and about the project itself, the NASA project, I'm going to talk about the technology a little bit. And the first question is, in the early 80s, people started realizing, for the longest time, people have said that the human eye is the best pattern recognition tool that exists. And it's true. We are extremely good at recognizing features because we grow in an environment that has features. So we have grown to recognize them. A computer is very bad at this. It's getting better, but they're not as sophisticated as we are. On the other hand, they're very good at quantifying things in a very reproducible manner. So if you go to the doctor and let's say you have to go get a biopsy, histopathologists will get something like this. It's an image, and you see those nuclei here, which is shown in blue, or actually it should be blue, but it's a dark staining here. And the nuclei, looking at this pattern, a pathologist will decide what type of disease you may have. Do you have a normal tissue or do you have an abnormal tissue? And they're very good at this. But now, the one thing they're not very good at, even though they're expert in recognizing pattern, is to quantify and absorb a lot of information at one time. And that's where computer becomes more powerful. So to illustrate this, that was done in the 80s. I'm just replicating what has been done in the past. Early 80s, someone challenged a lot of people saying, can you recognize the different patterns? So we regenerated those things. Here in those images, I created 5 different circles with 5 different diameters. So it's 5 units. And we mixed them in a random manner. And the question is, can you, with your eyes, decide if those 4 populations are the same populations, or in fact, they are, maybe there are some of them the same and some of them are different? And I would say, putting that to anyone, when people have been tested on this, it's really hard to tell anything. And in fact, in these images, there is 2 groups. So A and C are the same population, and they basically are, the majority of those circles actually have the median-sized circle, whereas the B and D actually have 2 populations in there. Not something really obvious by the eye, but it's really there. And this is what we're saying about quantifying biological images. I think this is the type of data that started really convincing biologists that they should go after quantitative tool, and they start involving a lot of money and development trying to quantify those images. So our work, we're not imaging specialists, we do a lot of imaging, but our work really is to quantify data as much as we can, image-based, and then integrate them into some models to actually predict behavior of human and how they respond to radiation. So fluorescence microscopy is a growing trend in the US, and actually in the world, I should say. If you look on Google and search fluorescence microscopy on publication, peer-reviewed publication, what you see is a big jump in the 90s. This is when the first fluorescence microscope became available, or I should say affordable. And there's a big jump where people start getting on the boat and start producing a lot of those images. And then since then, it's been going up nonstop. And the technology is getting better and faster. We have now broken the resolution of a microscope. It used to be 0.15 micron. Now we can go below that using 4-pi technology. So there's a lot of technology that's coming up around the corner, and we're generating more data, higher resolution, therefore a lot more. We need a lot more space. And just to give you an idea of the type of market we're dealing with just in the US, and just looking at the university, if you consider there's about 2,600 accredited universities in the US, and most of them have at least 1 imaging core facility. Each imaging core facility usually serves 50 to 100 people. You can immediately compute it's about 500,000 to 1 million people that deal with this type of images every day. And that's only in the academy. It's only in the US. So this is something that's growing, and it's just going to get harder. And basically what we start seeing in our lab, for instance, is we're getting a bottleneck in terms of how we deal with the data. We're starting to not process all the data because we just have too much. And we're not giving giving a good time on data. And basically we're not giving extracting what we can from those data because we just saturated. So The outline of the talk is the imaging challenges we've been facing over the past 5 years at the NASA projects. So NASA selected 5 universities, I should say 5 national labs in the US. And LBNL is one of them. So that we're given a lot of money to actually study the effect of radiation on human. We do one component of the project. And there's 5 other universities in the US that does that. And under the NASA project, I will talk about the effect of cosmic radiation on astronauts, really imaging-oriented type of talk. And on the imaging challenges, we'll go over how we sort and how we browse through our data right now, how we do segmentation, how we deal with registration, and how we deal with pattern recognition. And again, we're not imaging specialists, so if you're imaging expert, you might think our approach is fairly naive, but we try to, being a physicist, we're really talking to many different groups at the same time, and we try to integrate. We do quick imaging approach that gives us fairly quick answer, so we can link them back to the models and have results for the biologists. So it's kind of a weird niche where we are. So this is the type of data set we get. Usually you get a gallery mode where we can see as much as you can. It's really hard to see the data, but basically all those blue things are nuclei. They stand for DNA. And the different colors mark different things, such as DNA damage, for instance. And on those gallery mode, this is one software called Metamorph. We can go and click on anything, and then immediately the annotation of the image pops up. So we know what we're looking at. Then this is one where we have a quick view of our data. Then what we have is we use Matlab, actually. So we use many different software in the process. It's not something smooth at all. Under Matlab, Matlab is a great system to actually do a lot of prototyping. So we prototype a lot of our work under Matlab. And what we've been developing is tools to read different imaging formats, because microscopy, unfortunately, has different imaging formats. They're all TIFF-based. But then each company, because we're dealing with 5-dimensional images, there's X, Y, Z. Then you have channels. So the channel can be up to 32 channels at this point in microscopy. So you can acquire 32 different colors. So that becomes a dimension. And then you have time. So those 5-dimensional images becomes pretty hard to handle. And so they have created those embedded TIFF systems. And so Zeiss has created ZVI and LSM. Metamorph has created STK. Nikon has created ICS. And so we have to, each time, we have to go and read their special tags and have them under Matlab so we can parse. Because the beauty of reading those images and reading the information in the images, we can start sorting and parsing through things such as, for radiation biology, what we're looking for is the dose we gave to the specimen. So we want to know how much it got. We want to know when we stopped the specimen, when was it fixed. So the time it takes after radiation is an important factor. So the longer you wait, the other type of endpoint you're going to be interested in. The type of label you're looking at. So are you looking at DNA? Are you looking at DNA damage? Some proteins? The type of cells you're looking at. So all this is usually annotated in the image at the moment of acquisition and can be taken out. And then we also do one thing, classic thing is, we always do automatic segmentation of the nuclei. This is the one object that's always wanted in an image. We want to know where the nuclei are, individual cells, so we can then ask questions for each of those nuclei. So this is done on a batch mode as well. So nuclear segmentation. Nuclear segmentation, if you're lucky, you get an image like that. It's great, nice, uniform, homogeneous background. All you have to do is threshold it, and you get the contour. It's hard to see here. Now, that's a bit harder. Multiple nuclei, they're overlapping with each other, multiple intensity, multiple pattern. And so when we try to segment this, it becomes a bit difficult. And we don't have a great working solution. Nuclear segmentation is a field that's been done for, I think, 30 years. So there's many algorithms, and everybody claims they have the best system. So we're not in the business of doing nuclear segmentation. All we want is actually have something that is fast enough and that gets us through the data. And so those are different zones of problems here, overlapping, high intensity. So one of the problems is the background, for instance, variation of background. So in the same image here, you have a background that has a high intensity versus a low intensity. If you were to threshold this, if you threshold low, you'll miss some, or if you threshold high, if you threshold high, you miss those guys, but if you threshold low, you're going to start picking up stuff here. So you also have, within each nucleus, patterns that tend to be punctuated sometimes, and how you deal with this. So the approach is very simple. First, to go fast again, we subsample the image. We always do nuclear segmentation on a subsample image, because we don't need that much accuracy for identifying the object. So it's one little trick that we use. When we subsample the image, we can then pass it through a classic filter like Gaussian and median filter, which kind of like make the system more homogeneous. Then we threshold, get a binary mask. And then through an iterative process, we can actually find the seeds of the multiple objects in one mask. And I will tell you in a minute how we do that. And finally, we use watershed technique to regrow from the seeds, and we get our full nuclear segmentation. Interpreted back to the full size, obviously. So the iterative process is very simple. Like I said, there's many more advanced techniques, but that one is actually fairly quick. And what we do is we just do multiple erosion on a binary mask until we separate two objects. So if you do that on multiple level, you can separate, and it's, I'm not trying to claim we have the best system here. You can see how it actually didn't do such a good job. We separate that one very well, but we're missing some of those guys. And the reason is really, for something like that, you really got to go with a shape-based type of algorithm. Thresholding, and this is not going to cut it. But again, this is a very extreme case, and we have ways when we do the prep to minimize this type of events. So we can grow the cells and stop the cells at a moment where they're not touching each other, when they don't grow on each other. So the batch processing Matlab. So what we do with Matlab, then we can identify the nuclei, and we can also identify spots, for instance. So in this case, this image you're looking at here, those cells were traversed by cosmic ray. So each track here is a cell, is a DNA track. Those are DNA damage, those little dots. So what you're seeing here is human cells, breast cells. This is what we work on, breast cells. They're a great model for radiation because they tend to have cancer easily with radiation. So this is our model, human breast cells. And what we see when we put them on a beam, they create those tracks, and those are DNA damages. So we generated, first we get those huge gallery, and then we do automatic parsing to know what is what. So that image, for instance, got a iron particle. It was 1 gray, that's the dose, and that's 30 minutes after radiation. Then you can, so if I go back and forth, you can then segment the nucleus, and actually also you can identify the tracks. And those are label images, so you can identify different spots in the track. And then we can go ahead and do that over all our data. And if we go back to the plot I just showed you, the computer knows that this group, for instance, is a 50 centigrade, that one 100, and that one 300 centigrade. And then on the fly, almost, with Matlab, we can create a text file that will come and generate those graphs. And big surprise, what you see is what you expect, which is like the higher the dose, the more DNA damage you see. It should follow a linear trend. You double the dose, you double the damage, you double the number of damage. That's what you measure. Perfect. The slope itself is a very interesting factor, and that's what biologists are interested in. Why is that slope different than this one? Because this one was obtained with a certain type of radiation, that one with a different type of radiation. Each radiation have a different way of inducing breaks. So this is the type of endpoint that the biologist is very interested in having. They're not really so much into all what we've done, but they're very interested in this. So how does that link to Google? I think here, at least as a personal user of Google, the one tool that comes to your mind is Picasa, because if you install Picasa on your computer, what's amazing is it will go through your computer, sort everything, not based on scientific parsing, but scientific data, but still will parse and classify your data. And then you can browse through it. I think it's a great tool that doesn't exist in the scientific community. All the companies, Zeiss and Nikon, they have not developed a system like that. They're not very smart in terms of browsing through the data. And then in terms of searching through data, it's even more messy if you use one of those software, Nikon or Zeiss, because they don't really plan for this. So I think this is something I would love to see in Palimpsett's project, is a way to really do a lot of what we're doing and visualizing data in such manner. So again, it's a plug for my own interest here, but linking it to Google. So in terms of visualization, let me show you that. So again, visualization, we have to use a different tool. So we juggle with 5 or 6 different software. And each software actually is about 20, 30K. So we have only one software usually for the lab, and we have to share the machine where the software is on, because we just can't afford that many machines with those software. But so Metamorph, what we do is we just did nuclear segmentation, so we can do, and what I love about Metamorph is the interactive link between the data and the images. So for instance, here you can start gating your data. So this is the area of each nucleus in the segmentation, and they are visualized here. So you have a color system where you can say, well, I want to look only at the one above 100. They're showing green, the one under 100 are showing yellow. I can gate the data, I say I only want to keep those nuclei that have a size between 100 and 250, and here it is. And then after I can get, again, out of that population, I'm interested to see which nuclei are fairly round. So then I go for a shape factor. So I go click here, shape factor, and then I say I want a shape factor that's above 0.75 up to 1. And now I get the more spherical nuclei. This is just an example of how you go through an interactive, but I think this feature is really useful because it gives you a handle in your data very quick, and you can, there's that interactive mode between the data and the images, and you can really tune it. And that's where you use the best of both worlds. You use the best of the human because the human is so good at asking questions and at extracting pattern. And the computer is extremely good at giving it to the person. So if you have that nice dialogue between the machine and the human, it becomes fairly powerful. Now, one thing I didn't tell you yet is those images are actually three-dimensional images. So there's multiple slices. And so if we zoom on one of those nucleus, this is actually what it looks like. So again, that nucleus got traversed by, actually got 2 tracks. Well, I would say at least 1 track here. So that was 1 damage. We're looking at 2 different DNA damage proteins. So red and green are 2 different ones. That's why you have a yellow color. And then that's the way we visualize 3D images. We have that little cursor that you can move back and forth. And here I basically section the nucleus about that point. So that's the XY view. That's the YZ, and that's the XZ. And then I can, so that's BitPlane. So different software again. BitPlane can do very cool stuff. Like I can do a maximum projection by simply moving those 2 lines here. If I expand them, I can actually now do a maximum projection on my XY view. So if you go back and forth, you can see how this guy now shows up here. So there was another track here, actually. Okay, so it's a way to visualize the data. It's pretty nice, and visit the data. So other way to deal with those data is to actually do volume rendering. Volume rendering makes pretty images, but it's only useful if you can really play with the system and zoom in and out. So you can do isosurfacing and actually extract those spots using different color spheres. And you can make movies out of that. So, so that's the same thing now. And if it wants to work, I guess. I'm sorry about that. I think I'll pass on this one. Right, let's try again. OK, let's go. So I'll pass. Anyways, so what's the volume rendering of this and how you can visualize some data? So the last thing I want to talk about tools is pattern recognition. And that's something we're just trying to get into. If you see here, those are 3 different nuclei. And radiation tends to induce a change in the nuclear structure. We don't really understand it yet well. But you can do it with different things, like if you put salt in the media instead. So if you put the cells on the different salt, there's an osmotic reaction. And you can basically, under high salt condition, you will actually condense the nucleus. It will lose its water. And that will look like that. are under hypotonic solution, which is if you remove the salt in the media, the cell is going to start swelling, and it's going to look more like this. And that's a normal-looking nucleus. And so we wanted to see if we could, just by simply looking at the signature of intensity, see a difference. And actually, we were a bit disappointed to see that only the hypertonic tend to stand out. If once you have normalized the intensity, which you have to do, you normalize for the variance and the mean, you're going to see actually 2 peaks for those guys, for the hypertonic. But the hypo and the normal, really, you can't separate them anymore. So even though they are different. So clearly, this is a new project. We're not sure how far we're going to go with that. But obviously, what we're going to have to do, and that's what we've been talking about, is going with PCA analysis and using multiple features. So not only intensity, but also size and shape of the nucleus would be another feature that could be used to separate them. So anyway, so I wanted to cover all this. Now we're going to move to NASA projects. So I wanted to give you an overall view of the technology first. And so hopefully this will work. So NASA is interested in the effect of cosmic radiation on human. And the reason they're so interested is that in that animation, you can see those solar particles, actually they are protons, and hitting the magnetic field of the Earth. And actually what's interesting is the Earth is actually a tiny thing here. What happened is the Earth has that incredible magnetic field. It's really weak, but it has a huge range. And because of that, we don't see any of those particles on Earth. So the things I just show you, we don't have them on Earth. And so we have no data whatsoever on the effect of those particles on human because we never got hit by them. We have a lot of exposure to photons. The atomic bomb was mainly photons and neutrons, but nothing on cosmic ray. They don't come here. So because of that beautiful magnetic protection. So now you can imagine we're working for the mission to Mars. And the mission to Mars, they're going to have to leave this nice protected area. And even the astronauts right now working in orbit, they don't get that much compared to what they will get once they get out and go to Mars. They're going to receive a huge amount of those rays. And that proton is a predominant particle in space, but it's not the most dangerous one. The most dangerous one is iron. And iron is produced by supernova. And the supernova produce so much of that you see a lot of those particles hitting the human when they go on the mission to Mars. So we're very interested in studying the effect of iron particles on human. And so we do to get, where do you get cosmic radiation on Earth? Well, you got to go to an accelerator. And not many accelerators can produce the type of energy you see in space. So we go to Brookhaven National Lab in New York. That's the alternate synchrotron. And basically, NASA has built an extraction beam line, which is shown here. And then what we do is we go in the beam. So this is the control system. This is a whole key system with eye recognition so that when we go in the beam, we can't get shot. That would be better. We go in the cave. That's the cave. The beam comes from here. Those are our detectors. So we put a bunch of different detectors to quantify the amount of fluence of particle going through the specimen. The specimen is stored in an incubator, so it can stay nice and warm. And then you get an image like this. So those cells, human cells, again, got traversed by tracks. And the first thing we did is we wanted to actually identify the tracks. So what we did is initially we developed a technique where you can roughly draw on an image very quickly the nuclei you want to study and say, OK, the tracks went through here, here, and here. And then we refine the search, and the computer comes back with track detection. So the way we've done that, to give you an idea, is the following. It's a very simple idea. We do some projection of the specimen. So in this case, the gray image I just show you, if you project it in an angle that is not along the track, you're going to get a profile that looks like this. You get a few peaks corresponding to some of those spots. But overall, you get a kind of a so-so profile. Now, if you hit it right on the angle, and so I forgot to mention something, our beam is always parallel. So when we expose the specimen, all the particles are going straight. And they're so heavy and so fast. Their beta is 0.97, which means they're very close to the speed of light. And so they take about 12 centimeters to stop. So they go through our specimen without any interruption. And if you get traversed by them, they will basically pierce you right through. You don't sense anything. They're so small. But they all are parallel. So one thing that's good about our specimen is we know we're looking for parallel lines. So by doing that, when you do some projection, when you hit the right spot, you're going to see only a few peaks, and they're going to be very high peak. And again, if you use another angle, even in this case where you have those two guys, you're going to get a peak, but the peak will never compete with the right track. So by doing that, all we do is we do those different projections and keep the projection that gave us the minimum number of peaks and the highest peaks. And once you have that, you can locate the center and project them back on the angles, and you have the track. So that's how we generated those guys. So using those tracks, we had a great, so I'm a physicist by training, so I really wanted to extract something more interesting in the data. So one question, there's a lot of controversy about those proteins we're using to recognize those DNA damage. People say they are DNA damage, they're not DNA damage, they're more complex. So one thing we said is that, wow, one thing we know for sure is that at the moment of the damage, we know exactly by the physics what should happen. And what should happen is the following. If you get a track here, you're going to get a DNA profile. That's the blue color is the DNA. So you see, there's more DNA here, there's less DNA here. And basically what you expect to see is, on average, breaks should happen where there's more DNA. It's a trivial statement. If I take it to the extreme, no DNA, no damage. You can have a DNA damage if there's no DNA. So, and actually, if you do the physics, it's very simple. It's proportional. It's directly proportional to the amount of DNA. So knowing that, what we said is that what you expect to see on each track, if I get the DNA profile, I can actually, and I'm told here in this DNA profile there were 4 damages, I can take them and randomly distribute them on the track, on the profile. And if we do that many times, it's a Monte Carlo approach. If you do that many times, you throw them randomly following that profile, you should generate an average DNA damage profile that matches what you expect from the physics. And we did that using all that tools. And when we did that, we got those results. The blue curve is the distance distribution of those foci. So if you look at a track, there's different foci. And we're looking at the distance between each foci. And then we're generating those distribution. And what we see is the blue curve is what we measure. And what we measure, if we do the randomization, is a Poisson distribution. The Poisson is cut here because a microscope we're losing the resolution here. So we can't get the tail of the Poisson because we don't have infinite resolution. But that's fine. We still get the Poisson shape. But we get more like a kind of a Gaussian type of thing here. And the big difference is the predominant, the most favorable distance between 2 foci theoretically should be 0.3 micron, and actually we're measuring 1.2 micron. So what we're seeing is that those DNA damage proteins that we're using, the DNA damage are not where they should be. They have moved. And they have moved in a very interesting way. They seem to have excluded each other. You don't see that many close-by DNA damage. And this is getting worse 35 minutes post-irradiation. So that was a big red flag. When I presented this first in the community, they were like, no way. It's an artifact in imaging. And OK, so we had to convince them something was really going on. And so we really said, well, we have increased deviation from randomness, and there's an exclusion of close-by foci. So where are they? So I kept going on this one. And NASA gave me green light to keep going on this one. So I went on, and I said, OK, can we find out where they are? And we came up again with a very simple imaging tool. What we did is, I said, in the image, I can see that the break seems to be where there's less DNA, which is counterintuitive. They should be where there's more DNA, but in fact, in the images, visually, I would see that. So I said, OK, we're going to measure it. The way we measure it is, this is a cartoon. Well, this is a real nucleus image, but those little red dots, I placed them just to illustrate the point. And if you put those dots in a high DNA density region versus a low DNA density region, can I measure it in a quantitative manner? And the way you do that is simply by looking at the sum of those signals at those locations, normalize by the mean of the signal over the whole nucleus. And when you do that, when you're in a bright region, you're above 1. When you're in a dim region, you're under 1. Now, the other thing we notice is that a lot of those breaks seem to place themselves between the bright region and the dim region. Interesting. So for that, we took the gradient of the DNA, which gives you those kind of brain-looking like images. And actually, what you see is that that interface here has a high gradient. OK, it's classic. The gradient will actually will be bright where you have a change of intensity. And if you do the same type of computation now, you'll see a value above 1 when you are on those edges. So using those 2 parameters, Rdna and Rgradient, we went back to the images over that huge data set. Here we're dealing with, I think it's about 400 gigs of data right there, summarized in that one graph. So the 5 years is the number of experiments went to Brookhaven to repeat. So those are repeat. What you see is within the first 30 minutes after radiation, you have an R gradient that's above 1 and an Rdna that's below 1, which tells you that basically those breaks are happening not what you expect, because what you expect is that gray zone here. So they off the gray zone. But on top of that, we created the signature of where they are happening. And basically, if you see that slice here and that track, if I do a topographic map for the DAPI, for the nucleus, you see those, basically the higher, the more DNA, and then those valleys is where there's less DNA. And what you see is a lot of those breaks actually are in the valleys where there's no DNA or very little DNA. On top of that, what's interesting is those breaks are those at the interface between the high slope of those profile. This is just a visual of those, just to get an idea of what it means. And so we published that in PLOS, which is a fairly good journal. And what we concluded was that basically DNA damage are processed in some very specific region of the nucleus. And that's never been shown in human. And the imaging and the modeling was the key to come up with that. It's still a controversy. People still believe that there's got to be an explanation to our data. But now it's out there that we have at least released. Only yeast has been shown right now to be able to, when you have a damage, the yeast will actually cluster the damage into one place and repair. And that's a big deal for NASA, because if there is really damages being collected in one place, for gamma exposure, when you get a dose of gamma, for instance, you get breaks a bit everywhere. They're kind of sparse, so they will probably get repaired in individual repair center. When you get a high-fidelity track like that, I mean a cosmic ray, a lot of the damages are close by, so they're going to end up being close to each other and actually be repaired together. And guess what happened then? What happened is that the DNA gets confused, and when you repair DNA, you start putting one chromosome with another one. It's called translocation. And that's a precursor for cancer. Maybe that's the explanation why iron is so carcinogenic, because we know on mice studies that when you get exposed to iron, you get a 40-fold increase in cancer versus other type of radiation. So it is an ongoing debate, and we're still working on this. But one thing to solve the debate was to actually go for life-cell imaging. So now we're moving to 4-dimensional images. We have ways now using a green. So people have extracted from jellyfish the fluorescent protein that is fluorescent in the jellyfish has been extracted. And now we can encode it into a cell, and we can make the cell fluorescent. So what we did here, we took the protein of interest that was the DNA damage protein, and we fused it to the fluorescent, the DNA damage protein, and we made movies out of them. So we go, that's Brookhaven again. That's our microscope. That's the incubator where we keep the cells, and we image them here. That's the type of slide we're using, so the specimens are in there. And then I hope this is going to work. Let me see. I really like to show you that one, but if the computer doesn't want, let me see. And register. I don't know why I did that. But anyway, that's a nucleus after radiation, and it's moving. So the first thing we have to do is register the image. So the way we do that, well, for time purposes, unless you're interested, I will pass on the registration is done actually through a third-party software that we got on Matlab. So I don't have much to say anywhere on this. But once you have registered the image, he's going to ask me again for the direction, so bear with me. That one is DAPI register. So after registration, that's what it looks like. And so what I do to give you an idea of volume is we, under bit plane, we can rotate as we play the movie. So now there's no more movement, OK? So this is 3D registration, which is kind of heavy on the computer. And so now, foci reg spot. Unbelievable. All right. So now what you see is actually DNA damage movement. So this was kind of the key for us. We haven't published this yet. It's coming out because we want to do more experiment. But basically, what the biologists were saying that was impossible is happening, which is that they are really moving. We're not really sure exactly where, why, and that's what's going on right now in terms of the project. But what's really remarkable is that everything started with fixed specimen and using the heavy-duty imaging, processing, and all this quantification came up with this. And then we went back and we could justify enough to get more money to do this study on the live cell, which is always more costly to do these studies. So I think, again, and here this is a plug to Google again, but YouTube, for instance, has the movie compression serving online system, which is, I think, very useful for those 4-dimensional images. I mean, I'm not sure how I would deal with the third dimension, the Z, but usually A maximum projection is the way you do it. You do maximum projection, you get a 2D movie that way. So there's ways around, or you do volume rendering. So that's why I was trying to show you the type of what it looks like on the commercial package we use right now. This is the type of images we get. And I think, again, this would be nice to integrate in palimpsest eventually if we want to look at those data. So two last slides. Basically, one thing I think also that links nice to Google is, and that's really more in the future, I think, but one thing we do on the microscopy is we can acquire the low magnification of specimen and then revisit the specimen at a high mag. And that's very useful to get an idea of what you're dealing with and then pinpoint towards region of interest. So what we can do is we can actually store those large low magnification images on a server, and actually we are able to pull out from the freezer the specimen, put it back on the microscope. We can home the stage, and can basically, with the coordinates, I can go back to the same specimen. And if I'm given the coordinates, I can go and re-image this exactly the same way. So with that in mind, I think it would be amazing to actually have some type of low magnification storage of specimen, have people, biologists, go on their computer, visit the specimen, and then ask, OK, I want to go back and pinpoint region of interest where you go back and I do a high mag. And if I show you that, so again, those are human breast cells. The project, the reason why LBNL has been selected by NASA to do those studies is because LBNL is expert in growing human breast cells. And not only we grow them on a dish, we can grow them in a gelatin-like. And so what happens is when we do that, we can grow the cells and they make those aggregates. And those aggregates are like mini breast duct. And actually, if we give them hormones, they will secrete milk, human milk. So they are. So as far as we're concerned, that's the best in vitro system for humans to study radiation, because we're looking at a functional organ, a mini organ in a gelatin that actually works exactly like the breast, just at a smaller scale. So this is the reason why NASA was so into having LBNL working on this, because we've been using this type of system. Now, if we focus just on the imaging part, those big blobs here are like many, many cells, and inside the blob there's milk. But what we can do is people are interested in those features here, those kind of like branching. So that's a low mag image, 10x. I can go on the computer and visit that image, and this is actually the montage. If I pull out the full resolution of that image on 10x, this is what I get. But now what we did, having the coordinates, I went back to the specimen that was in the freezer, take it out of the freezer, and I say, OK, I want a 40x image in this region. This is what you get. So again, really, there's a lot of things that are still out there that are still not done, and I think that the scientific community will benefit a lot by some type of collaboration. And I wanted to conclude on, I think we need to integrate better acquisition and data storage, and that's the reason why we're so interested in palimpsest. Also, the one thing to remember is that the storage solution should host eventually good visual sorting and organizing capabilities, and I think this is what Google is really expert in all those levels. So visualize 5D images might be the one thing that Google is not so expert in. It's kind of a special feature. The basic tools, registration, segmentation, I am pretty sure you have better tools than we have. Interactive link, I hope I convinced you it's a very nice way to go back and forth with your data. Now, one of the annoying features is the fact there's multiple formats. They're not very hard. Some of them are actually, you would need agreement, or we need a special agreement with the company to let us access the data. It's non-NDA. We have to sign those things. So I'm not sure exactly how it would work. But anyway, multiple microscope format is actually a problem right now. And obviously, the one thing that you really need is the metadata so that you can parse and actually generate on-the-fly graph, like I show you. So kind of like data spaces you were mentioning. This is where it would fit nicely. And I think one place where everything is done would be great. And to give you an idea, what I showed you today was made with 5 different software. It's not as smooth as it looks like. And it's actually one of our expertise is to know what tools is out there and what we can use. And so, of course, expanding our toolkit would be great. The software are very costly. And also, when we go from software to software, there's no API whatsoever, except for BitPlan, where they created a plug from Matlab, which is great. So we can call the function and actually have them talk to each other. Except for that one software, the rest is you're on your own. You go Visual Basic, or you go with some type of macro. Usually, it's more macro. And no API. So once you leave the software, it's saved under their format, and then you got to reread it into another. So you duplicate data left and right. It gets really, when you have gigs of data, it's really annoying. So again, a place where everything is would be the ideal situation. And acknowledgement, the chair of the NSCORE project, NASA Specialized Center of Research, Dr. Barcelosov. Those are my collaborator at NASA. Francis Cucinata is actually the director of the project. He's the one funding all the different projects. And he's very interested in our data because he's doing a lot of risk modeling. So he's using our data to do risk modeling. And people at RBNL. And if you have questions or want to talk more, be glad to talk to you. Yeah.
What kind of problems do you think would be when you have imaging that you cannot make new imaging of, kind of like if you have radiation over time, looking at older situation in the same specimen, etc.?
So I'm sorry, the question is if the imaging looks different, how we deal with it?
No, if you have imaging over time
Mm-hmm.
and how to look at, well, how do you handle older imaging if you want to take a look at more details, then you would have kind of like enormous amount of imaging.
We do. We actually store all of them, and we usually have a time course that goes from 0 minutes, 3 minutes to the first hour is fairly well covered. We have about 5 minutes, 10 minutes, 20 minutes, 30 minutes, 45 minutes, 1 hour. And then as we go further in, we start changing the scale. And we go up to 48 hours. And then for other assay, then we have different questions because looking at DNA damage, they're all gone by 48 hours. They have been repaired or destroyed. The cells have died. There's a process called apoptosis where the cell will actually kill itself if it's healthy. So, but we store everything. So this is actually what palimpsest is going to be storing is all those data. So we could go back and the real problem right now is we don't have the manpower to. We store all those data, but we only can look at specific endpoint and at a certain limit, and we have limitation what we can look at. So if you could come back and say, you know what, we should look at this, then it's really hard for us to restart everything. You got to pull out the nuclear segmentation again, and because it's not usually eventually our data are archived, so you got to pull them out of the archive and a few things like this. That makes it. Did I answer your question? No. Huh?
Almost.
Yeah.
Yeah. The other thing is, what amounts do you have at this moment?
What in terms of data right now? So we have generated about a terabyte. So it's. And a lot of it is duplication from all those processing. So, and I think on palimpsest, what we plan to put more is like 200-300 gigs, I think. The lifestyle imaging actually is takes a lot of memory because now you're looking at it. If you do Z-stack over time, it gets really bad quickly. Any other question? No? Okay. Thank you.